Hello, welcome to single-cell multiome analysis series with SciDAP. In the previous video, we started ATAC-Seq Dimensionality Reduction analysis, so let's quickly review what we have here. In the QC tab, we see the basic QC metrics. Here on these U maps, ATAC fragments in peaks, the peaks themselves, transcription start site enrichment score, nucleosomal signal, fraction of reads in peaks, and blacklist regions. We see that everything is uniformly spread, more or less. Then we switch to the next tab, per dataset. There you could see all the ATAC metrics split by each sample. You could see our datasets and the clusters that were produced by this pipeline. We don't know yet what these clusters mean yet. It will be in our next videos. Here again, ATAC fragments in peaks for 4 levels: low, medium, medium-high, and high. This is peak per cell and other metrics that were shown before. Then the same thing, but per group. Interestingly, that as in the case of our RNA, we could observe that ATAC peaks for our TRM group is not uniformly distributed between every cluster. On the last step, when we will be reviewing the output of W and N clustering, we may look into this closer. Again, this is all the same metrics, but for the both groups. And their files with all the PNG plots and the workflow. And this is pretty much it for this intermediate step, dimensionality reduction. And in the next video, we will start single-cell ATAC clustering. Thank you for watching.